afternoon, everyone, and welcome to the Public Health Practice Grand Rounds for June 2011. My name is Molly Mitchell, and I'm the coordinator of the Mid-Atlantic Public Health Training Center. And on behalf of the Center and the Department of Health and Mental Hygiene, I'd like to welcome everyone to today's Public Health Practice Grand Rounds on community-associated MRSA, sources and pathways of exposure. Before we begin today's presentation, I'd like to invite everyone who is watching online to please visit our website and look at our available online and face-to-face trainings. In addition to the trainings available on our website, such as public health nursing and many archived grand rounds, our upcoming trainings include next month's grand rounds on home births and the public health response, promoting informed choices and healthy outcomes with Dr. Eugene DeClercq and Mari Breen Rothman, and the August Grand Rounds on public health and the alcohol tax campaign, how Marylanders beat the alcohol lobby and why it matters, with Dr. David Jernigan and Vincent DeMarco. For those watching online, we also invite you to email questions for our presenter at any time during today's broadcast by just clicking on the link right there on your screen. And we also ask that all of you please sign in so that we have a better count for our federal funders of who's watching today. And with that, I'm going to go ahead and introduce today's presenter, Dr. Ellen Silbergeld. Dr. Silbergeld received her PhD from Johns Hopkins University in 1972 and completed a postdoctoral fellowship in neurotoxicology at the Johns Hopkins University School of Public Health and an NIH staff fellowship in the Behavioral Neuropharmacology Unit. Her career includes a senior scientist position at the Environmental Defense and professorship at the University of Maryland School of Medicine. She has served as a scientific advisor to the Center for Disease Control, the EPA, National Toxicology Program at NIH, the Department of Energy, OSHA, states of Maryland and New York, the World Bank, ILO, United Nations Environment Program, and Pan American Health Organization. Her research and professional activities bridge science and public policy and include epidemiological studies and mechanistic research on gene-environment interactions and movement of pathogens in the environment. Some of this research is conducted internationally in the Amazon, Mexico, Mongolia, Thailand, and the Netherlands. Dr. Silbergeld's honors and awards are many and include fellowships from the Fulbright, Kennedy, Rockefeller, Danforth, and Woodrow Wilson Foundations, Phi Beta Kappa from Vassar College, the American Public Health Association's Barsky Award, and the MacArthur Foundation Genius Fellowship Lifetime Achievement Award. She is a professor of environmental health sciences at the Johns Hopkins Bloomberg School of Public Health. And with that, I present to you Dr. Ellen Silbergeld.
Thank you very much, and welcome to this presentation for the Grand Rounds for the Mid-Atlantic Public Health Training Center. I'm going to focus on one of the most important public health problems today in the United States and around the world, which is associated with exposures to the drug-resistant bacteria, methicillin-resistant Staphylococcus aureus, or as it's usually known, MRSA. And what I'd like to explore with you are the issues around the sources and pathways of exposure to community-associated MRSA. So I'll try and get us to that point of where we are in this problem disease and why community-associated risk factors are now extremely important for us to understand in public health. So methicillin-resistant Staph aureus, which is shown here in a stained culture slide, is the leading infectious cause of morbidity and mortality in the United States today. It has surpassed tuberculosis and HIV. So for this reason alone, it deserves the attention of public health practitioners. It's usually associated in terms of disease with skin infections, but it can also cause toxic shock syndrome and organ system diseases, particularly if it enters the bloodstream. Increasingly, MRSA is a disease of children, and this is a relatively recent change in the population prevalence. Moreover, it's important for us to remember that it's not only resistant to methicillin and other beta-lactam antibiotics, but it's often highly resistant to many antibiotics. So it may be more appropriate for us to refer to this as multi-drug resistant Staph aureus instead of by this one indicator antibiotic which brought it to prominence. Well, to kind of keep the notion of a grand rounds, let me start by framing this talk in terms of a case presentation. And this arises from some actual research projects conducted by Dr. Amy Peterson and Dr. Megan Davis in my laboratory and ongoing work in our group. So from this, let's consider a case presentation in the classic mode. Jane Doe is a married 45-year-old woman residing in Lancaster County, Pennsylvania, and she works at home as a telemarketer. She has no recent hospitalizations and no reported recent infectious diseases. Upon entering a local hospital for elective surgery, not related to any infection, she presented at a major teaching hospital in Pennsylvania, and in accordance with state guidelines in Pennsylvania, it's recommended that all patients who are admitted into hospital are screened for carrying MRSA. And this is done, as you can see in the lower picture, by a nasal swab. This is not infection, it's really an indicator of exposure. However, it's been shown that people who carry MRSA are 4 times more likely to become infected by Staph aureus or methicillin-resistant Staph aureus than persons who do not carry the pathogen. Upon this testing, Jane Doe tested positive for MRSA. Now, why was she tested and what are the consequences? She didn't present with any signs or symptoms suggestive of MRSA infection, as I indicated before. She didn't report any recent contact with healthcare facilities or other common risk factors, and we'll spend some time talking about that topic. So why was she tested? Why does the state of Pennsylvania and several other states have these recommendations? Primarily to control infections once people come into the hospital, because there has been an increasing problem of MRSA cycling in healthcare facilities and then endangering the health of a large number of people, particularly people who may have compromised immune systems or undergoing procedures where the integrity of the skin and body are interrupted, such as surgery. And there are tremendous costs associated with controlling these hospital-based infections. What's the response to Jane Doe or anyone else testing positive at admission? Well, in most hospitals, this will activate increased infection control measures because the results are received usually within 24 hours. Remember, the screening is done at admission, and the results are available the next day. It may trigger retesting to ensure that the patient herself does not become infected with disease, and there are now some projects ongoing to determine whether intervention should actually be directed towards being positive for carrying MRSA, even in the absence of infection. MRSA is usually diagnosed by clinical signs and symptoms, and I'm showing a kind of classic presentation of a skin infection on the leg of this particular person, and the call or the differential diagnosis that it's actually Staph aureus is done by collecting samples at the site of an infection or from the nose, as I mentioned before, to detect carriage or exposure. And in standard microbiologic methods, the culture is then prepared through a set of selective media and other processes. The phenotype or the appearance of Staph aureus is confirmed, and then there is a direct identification, usually done by genetic screening. In addition, at this stage, we can distinguish whether this particular Staph aureus is susceptible to methicillin, that would be MRSA, or is it resistant to methicillin through exposure in vitro. We can even go further and start to get an idea of which strain of MRSA has been isolated from the patient through a variety of specific genotyping techniques. And these will be important as I talk about the search for the community risk factors. And there are three general ones, either looking at a specific gene, that's the MEK gene, which actually confers resistance to methicillin, or to SPA gene, which may give identification of the strain of Staph aureus independent of being resistant to methicillin, and also looking at a range of genes that encode for virulence. Or we can even start to type longer stretches of DNA, going so far as to do whole genome screening in order to identify which particular organism is present. And we know in terms of surveillance and detection of origin and spread within communities or within institutions, for example, from 10 or 20 years of research on tuberculosis, that this is a very powerful method of uncovering the source of a pathogen within a population. And this just gives you an idea of the different kinds of data that are generated in terms of both phenotype, that is resistance patterns, in terms of general genotype by pulsed field gel electrophoresis, and in fact by specific analysis shown in the lower center, and that's of the specific MEK gene cassette, and even of a broader genotyping analysis as shown on the lower right-hand side. You can see that really MRSA has become, as CDC has called it, an epidemic within the United States and many, many other countries. So over a relatively short period of time, a geometric increase in the prevalence of carrying MRSA at hospital admission has occurred between 1995 and 2005, and it has not significantly decreased. But it's not only increased over this period of time, it's changed. And this is really the heart of why this becomes a very important issue for public health, is to recognize the changing nature of MRSA, moving from healthcare into the community. Now, what this slide shows you is, again, roughly that same period of time from 1995 to 2005, and these are data collected from a range of hospitals by CDC and integrated. The blue line at the top shows the number of unique cases of Staph aureus that are isolated within hospitals, and you can see that that line really remains pretty constant from 1996 through 2001. There's not really a change in the number of cases that are being reported. The red line underneath that is all MRSA cases. You can see there is a gradual increase over that period from 1996 to 2001, but it really takes off after 2001. So let's explore what's driving that. The healthcare-associated MRSA is the green line, the pea green line, that is also flat. So it's not an increase in MRSA associated with healthcare institutions or risk factors. That hasn't changed. What has changed, the purple line and the blue line. The purple line are what we call community-associated cases of MRSA, and the blue line is one specific strain found in community-associated cases known as USA 300. So those are really the drivers of the great increase in MRSA cases in the United States. So it's not a change in Staph aureus, it's not a change in the healthcare-associated strains, it's really a change in these community-associated strains. It's also, as I mentioned at the outset, becoming a disease of children. These are some data that were compiled again by CDC, again over that same period of time in which they used the National Hospital Discharge Survey data and others to generate. And what you can see that, again, about 2001, there seems to be an uptake in the number of cases that are reported, and this is now actually a prevalence or an incidence of newly diagnosed cases among pediatric patients admitted to hospital per 100,000. So it has a baseline denominator as well. There's also a very interesting difference by geographic regions, and you can see the greatest increases and the greatest incidence, almost between 2 and 4 times as much, is actually occurring in the southern region of the United States. Again, community associated, perhaps now some geographic differences in how this new presentation of MRSA is actually presenting itself. Well, let's get to some definitions. I've been throwing around healthcare associated, community associated fairly loosely. And the most important, or one of the important messages of this talk really is that MRSA is not just a healthcare associated disease. That was 10 years ago. Since then, as I've shown you, it's predominantly driven by the increase in community associated strains of the pathogen. What do we mean by that? Well, CDC, clinical care, hospitals, and others came up with a fairly good definition of what they meant by healthcare associated back when this was considered primarily a nosocomial or healthcare origin condition. Healthcare associated risk factors included employment in or recent contact with healthcare as an inpatient nursing home or any other facility which could be defined as healthcare associated, recent use of antibiotics, or diagnosed after the person had admitted to a healthcare setting. So the person comes in clean and then acquires the disease. Community associated is kind of this unknown planet. It's sort of not healthcare associated. So it's present at admission to healthcare. Obviously, then it preceded the contact with healthcare, but we don't always get that information. Screening for MRSA is not mandatory, nor is it really practiced very widely throughout the country. And you can see the other defining characteristic, these come from CDC, these definitions, is that there is no healthcare associated risk factors. It doesn't give us a whole lot to go on related to the major burden of illness of this pathogen. This sort of definition by negation. I'm also going to spend some time on a new category of MRSA, which is called zoonotic, which means that its origin is within non-human animals. And many of our infections and pathogens, particularly bacterial, have originated first within non-human animal populations, and our contact with them, our consumption of them, our residence near them determines the transfer from the animal populations, along with genetic changes often involved, into the human population, and then transmission from humans to humans. So the classic definition of any zoonotic infectious disease would be that it involves close contact with or residence near animals. And in this case, it's primarily livestock. Now, these differentiations here are being made classically by sort of epidemiologic and clinical individual risk factors, but they can also be done, and people are very interested in trying to do this, by genetic analysis. So there's certain genotypes, certain strains that are associated with healthcare sources, with community sources, and possibly with zoonotic sources. To some extent that's true, but for reasons I think you'll understand, this becomes harder and harder to differentiate because we all live in kind of a mixing bowl of ourselves and the pathogens we carry. And sometimes also done by analysis of the types of antibiotic resistance patterns that they present. But it's still primarily by risk factor. So let's try to unpack this major category of community associated, because that's been a large part of the work in my research group has been to understand what is this? What are the risk factors? Well, it's diagnosed in community settings. That's by definition. Its prevalence has been related to identified risk factors, for example, crowding. So there have been outbreaks or increased prevalence or incidence among military populations, populations in prisons, populations in schools. And we know that certain practices that probably both are associated with decreased integrity of the skin, and as well as general unsanitary conditions and risks of injury are also seen as risk factors. For example, like my son, being the member of an athletic team was also a great publication from MMWR among persons who went to a tattoo parlor, which of course is where you're getting your skin broken open. But this is not where it's coming from. These are the populations at risk, but this doesn't tell us why are these new strains, relatively new strains over the last 10 years, appearing within the microbiome of Staph aureus. We can understand why these groups are at risk of having infection or even exposure, but this doesn't tell us really where the pathogen is coming from. What are the origins of community-associated MRSA? Why is this important, and how can we explore this question? And that's really what I want to talk about in this talk. So let me give you some terms which are used in sort of hunting for community-associated MRSA or any other emerging pathogen. So we're interested in resistance genes because they encode for molecular changes that confer the phenotype of being resistant to an antibiotic. And these genes often travel together in what are called cassettes. Bacteria possess the extraordinary ability to transfer genetic information from one organism to another without having to reproduce, although they do that pretty efficiently and rapidly as well. But mature bacteria can actually send genetic information from one organism to another, which means that whatever information is available within the community can be widely dispersed. And so increasingly, certainly, when we start to think about this in terms of the environment, and the environment as the domain in which community-associated MRSA is occurring, both arising and moving around, that we're really driven to adopt an ecologic perspective. So we also think about the microbiome, which is the entire microbial community within a specific region. Could be your gut, could be your skin, it could be this building, could be a farmhouse, it could be the water supply, because this is what is available. And within that microbiome, a term has been coined called the resistome, which is all the genetic and other information that's available to all the members of this microbiome, all the microbes, that empowers microbes to resist and respond to antibiotic pressure. So this is the context in which we've tried to design research to look at and answer those questions. What is community MRSA and where is it coming from? Well, let's step back and talk a little bit about antimicrobial resistance in general, not limited to methicillin or to Staph aureus. And we've known for a long time what some of the determinants of increasing antimicrobial resistance are related to. And certainly it has to do largely with how we use antibiotics, the drugs themselves. As you may know, most antibiotic or antimicrobial agents are derived from natural products, from other bacteria, from fungus, or other microorganisms. So the kind of, if you will, competition between bacteria and the other microbial life within soils and water, long before we were on this planet, has been going on for billions of years. And over that time, processes of natural selection have, of course, resulted in the fact that bacteria have obtained, through spontaneous mutation and then pressure, advantage, that is, the genetic machinery to resist all these natural compounds. So nothing new is being created here, and that's our problem. We really have very little that is new. Unfortunately, we use antibiotics in a very indiscriminate and uncontrolled way. Even though back in 1944, when Alexander Fleming won the Nobel Prize for penicillin, he warned against this. It's little known fact that within 3 months after he began to apply the isolated form of what we now call penicillin to his bacterial cultures in the lab, within 3 months he observed they were becoming resistant. So he knew already that this was a gift, if you will, that needed great control and management. We've done the opposite. It's been used in agriculture, used in consumer products. I'm sure you know all about this. There is clinical misuse and overuse of antibiotics, patient compliance in terms of how individuals use antibiotics. And increasingly, and not just in the developing world, there's a lack of control in terms of access to antibiotics. You can go to pet stores and buy antibiotics. You can give them to your children, to yourself if you would like to do that. You can buy them over the internet. They are widely available. So we've not done a very good job about controlling this driver, this cure, and also this driver of antibiotic resistance. So, as I said, this is because the bacteria already possess the genetic machinery to respond to these drugs, and that antibiotic use is associated then with selection for resistance, but also with increased rates of exchanging genes. So not only is there a pressure so that the organisms that carry a gene conferring resistance survive, that can be shared within the community. And a number of studies have shown on a national level, a population level, or within a particular hospital setting, that the intensity of antibiotic use is directly related to the likelihood of resistance and the prevalence of resistance. And inappropriate antibiotic use selects and disseminates resistance because it's more permissive of the survival of even partially resistant strains. So one of our questions, and it might be, is in order to understand where is community MRSA coming from, where are we using antibiotics? Where are the antibiotics in our society? Well, we think they're in hospitals, right? Or we think they're in our medicine cabinets. But you would be wrong if you thought that. Most antibiotics in the United States are fed to animals. These data were generated by the U.S. Food and Drug Administration earlier this year from a call-in to the industry. Now, this issue has been highly controversial for about 6 years because the industry would not reveal the percentage of their products that were going into animal feeds. The agriculture industry wouldn't tell us exactly the poundage or the tonnage of antibiotics that they were using. But the FDA, and this was done by Dr. Sharfstein, actually had the power to require the companies that made the antibiotics to answer this question. And what I want you to just notice is that at the top, this is comparing human clinical medicine to the use of antimicrobial drugs in animal feed. So this is not veterinary medicine versus human clinical medicine, okay? Human clinical medicine accounts for about 20% of the total usage of antibiotic drugs in the United States. Use of antibiotics in feeds accounts for 80%. So if you want to know where the antibiotics are, they're in animal feeds. And that's the red is the animal feeds. And you can see from many drugs, the majority is actually going into animal feeds. And the small amount that is in human medicine would require an explosion of this scale for you to see it. So almost every class, every molecular class of antibiotic drug is overwhelmingly devoted to use as a feed additive, and the total picture of antibiotic production says this is where the antibiotics are. So this provoked some of us to think, well, if this is where the antibiotics are, maybe this is where the antibiotic resistance is starting, particularly outside of the clinical or healthcare setting. So I'm not going to spend a lot of time on this topic, but what is this with antibiotics in livestock? Well, it's part of the whole transformation of food animal production, which has gone from this top picture, which is, by the way, what Purdue would like you to still think they have in terms of raising billions of chickens, to the bottom picture, which is intensive swine production. So it's highly intensive, confined. The animals, whether they're pigs or poultry, are confined for their lifetime within buildings, highly regionally localized. The whole goal is to generate a high throughput. This is accomplished through highly engineered feeds. These animals aren't eating forage feeds or their natural feeds, but a powdered production that's primarily soybean and corn based, to which a number of things are added in order to enhance growth and suppress certain diseases. The environments are controlled, and this results then in a standard product which can enter really an industrial production line known as the slaughterhouse. All the problems that are related to this are outsourced from the industry, so we don't even know about them. Just another way of looking at this dominance. An analysis that was done by my colleagues at the Environmental Defense Fund looked at the estimated use of antibiotics And this is looking at feed additives in swine production as compared to human medicine. And what I just want you to take away from this is just another way of showing the enormity of the prevalence or predominance of this feed use as compared to human clinical medicine. On the right-hand side of that graph, you see the human clinical use for the entire nation of the United States at 3 million pounds per year. That is exceeded by the use of antibiotics in just one state, North Carolina, for the production of pigs. Another way of saying, if you want to know where the antibiotics are, this is where you really need to look. And on the right is a wonderful symbolic picture drawn by the Hopkins art department describing some of our work, which shows a chicken stuffed with antibiotics, which is, I have to say, how I think about this right now. This is really what we're eating. We're not necessarily eating the drugs, but we're eating the results of this massive use of drugs. Well, why would this really be associated with MRSA? Well, first off, Staph aureus is a very common pathogen found in domesticated animal species. Very common for us, very common for animals. And it's been known for a long time that veterinarians and farmers are known to be at increased risk of exposure and infection to Staph aureus. But why MRSA? Well, we know, as I've just pointed out to you, that livestock, pigs, cattle, poultry are exposed to antibiotics, and therefore there's strong pressure for the selection for resistant strains. But let's go one step deeper. An analysis has been done now to start looking at this. Well, since it looks as though this is where the antibiotics are, is this where Staph aureus and MRSA is? And a large number of studies that have been carried out predominantly in the European Union, but also in Canada and the United States, have now reported a specific strain or specific strains of methicillin-resistant Staph aureus in animals. So much so that a number of public health officials are now suggesting that we really do need to think about certain strains of MRSA, perhaps the dominant strains now, as zoonotic in origin. This has big implications for public health response and the importance of integrating both veterinary and human clinical medicine, the one medicine approach, in terms of understanding this disease and controlling it. The first real demonstration of this, I think, was in this very famous study conducted in the Netherlands. And these are two graphs produced by colleagues in the Netherlands. And what you see here are two maps looking at people and pigs. Now let's go over to the right-hand side. And what you see here in the various shades of pink and red are the density of people in the Netherlands. And the darker areas are, as you expect, Amsterdam and Utrecht and The Hague. And the lighter areas are the countryside areas. And here they looked at all the reported cases of hospital-associated MRSA within a period of time in the Netherlands, and they found that this really did cluster where the people were, which is not surprising. But at the same time, they had all these other cases of MRSA infection that weren't identified as being one of the hospital strains. It's before we began to have the genotypic data identifying some of these community strains. So these were known as non-typable. You can see they're not in the areas where the people are. So if you look over at the coast of the Netherlands on the left-hand side of the slide on the right, you can see those don't appear where the people are. And then, very interestingly, they decided to say, I wonder if it's appearing where our major livestock production animal is most dense. And so on the left-hand side, what you see now with the varying shades of red and pink is the density of pigs. And this was the first report to suggest that actually this might be associated with pigs. Now, Beth Feingold, who's recently completed her dissertation in our group, went further in terms of a highly sophisticated spatial analysis of these data and reported not only is it related to direct contact with pigs, or the number of pigs, but just living in an area with many animals increases your risk of carrying these community-associated strains. But this was the first real epidemiologic signal that we had. So it's not just working with livestock, and we can think of ways in which livestock-associated MRSA, which is what the Europeans now call this subset, if you will, of community-associated MRSA, which, by the way, is about 35 to 40% of all community-associated MRSA infections in the Netherlands and Belgium. So, people who are raising animals and transporting animals and working in food processing plants are members of communities. So they're interacting with the community, their children are interacting with the community. These facilities are releasing materials all the time. That's been a major area of study in our group. And these people go into hospitals. And this gets back to my comment earlier that it's very hard to kind of now talk about a pure healthcare-associated strain or a pure community-associated strain, because you have people from the community who may have picked up a livestock-associated strain, say in a farmhouse there, who now go into the hospital. And now this becomes part of the microbiome, if you will, of the hospital. And it's much more fluid in terms of the movement of both people and bacteria within these systems and environments. It's a problem for us. I want to focus your attention to the fact that we live within an area which constitutes some of the highest production areas for both poultry and pigs in the United States. And I've circled that. This area here is showing you poultry. Some people refer to this as the broiler belt, which is somewhat coincident with the Bible Belt, but I won't go there. But you can see Maryland, the state of Maryland, which is very, very dense and historically has been one of the most intensive regions for the production of broiler poultry in the United States. In fact, the two counties with the densest numbers of chickens per square mile are in fact in Delaware and Maryland. So we are still retaining that. North Carolina, you remember from the data about the antibiotic use, is the number one or number two. They're always in a neck-and-neck race with Iowa for swine production. So the Mid-Atlantic, this is an issue we need to attend to from an environmental perspective as well as an infectious disease perspective. So now that we've kind of established that part of the community-associated MRSA epidemic may in fact be zoonotic in origin, whether we call it zoonotic or livestock-associated, probably we should start calling it zoonotic because some of these strains are being identified in cats, in dogs, even in hamsters, so that by restricting our attention to livestock, we may be omitting other opportunities for contact and transmission. What does this suggest then about points where we ought to be concerned of risk of exposure? Well, certainly I've already mentioned that persons who live near these areas, persons who work with livestock or with animals have been shown to be at high increased risk of both exposure and infection by these zoonotic strains of MRSA. Let's take this a little bit further because these changes in the distribution of MRSA from healthcare to community are national in scope. They're not restricted to these areas. And so therefore, we can't restrict our own thinking about the implications of this information. What about people who eat meat or poultry? Now we know that food is a major conveyor of pathogens. Foodborne illness is a major component of infectious disease and, in fact, illness and death in the United States. So what about MRSA and food? This is something nobody looked at until very, very recently. One very recent study done by Dr. Lance Price, who recently received his PhD from Johns Hopkins, was a national examination of Staph aureus isolated from a whole range of consumer food products bought at consumer outlets, grocery stores, almost all national, large grocery stores throughout the United States. And I want to direct you to both parts of this slide. The overall findings are on the right. What they did was buy consumer products sealed at the plant at these retail outlets of turkey, pork, chicken, and beef. And what they found, as shown here, is that of the turkey samples, 77% of them were carrying Staph aureus, 42% of the pork, 41% of the chicken, 37% of the beef. Now, 96% of all these isolates were resistant to at least one antibiotic. It wasn't always methicillin, so you wouldn't technically call them MRSA, but they were resistant to an important drug. 52% of all those isolates were resistant to 3 or more drugs, which is the CDC FDA definition of multi-drug resistance. And those patterns are shown over there on the left in terms of resistance. And then this involves erythromycin, oxacillin, tetracycline, penicillin, ampicillin, levofloxacin, ciprofloxacin, and then, of course, multi-drug resistance. So this is a pretty daunting picture if you think about one of the implications of having a zoonotic pathogen is the potential risk to food preparers and to food consumers. And as I said, we know that food consumption has been associated with multi-drug resistant infections. This is one of the more disturbing studies, I think, in which a study was conducted, a case control study was conducted using women who had UTIs, and from these infections, E. coli was isolated, and the isolates were then tested for a range of antibiotics in vitro, and then they were compared to see whether those women who tended to have resistant E. coli were more likely to have consumed poultry. And what you can see is that, in fact, that the odds of carrying multi-drug resistant E. coli were almost 4 times higher with recent reported poultry consumption as compared to those who did not report recent poultry consumption. So the role of contaminated food in drug-resistant infections has certainly been demonstrated in a number of studies. And we do generally have a problem in the U.S. food supply, not just MRSA. This is a survey that was done by the FDA, and this is looking at antibiotic-resistant bacitracin, ciprofloxacin, erythromycin, tetracycline, and a range of meat products. And you can see these are pretty high prevalences in terms of carriage of resistant enterococci on these products. So when you go to the store, be very careful. When you unwrap your chicken or beef or pork products, be even more careful. When you prepare it in your kitchen and put it down on the counter or the cutting board, be very, very careful. And when you eat it, make sure it is very, very well cooked. So let's go back to our detective work. We know that there's a lot of antibiotics present. We know there's staph present. But why would this be related to MRSA? And what might be some of these stealth drivers of resistance, particularly related to community MRSA? Well, we know bacteria reproduce rapidly. They can share genes. Remember I talked about horizontal gene transfer and the presence of cassettes or groups of resistance genes. Well, the gene that confers resistance to methicillin, known as the MEC gene, is usually found on a cassette, that is, a set of different genes. And often this cassette contains genes encoding for resistance to other drugs, as you saw from the results of the study by Lance Price. Many isolates of livestock-associated MRSA are also resistant to tetracycline, and that was also shown on that slide if you saw it. And tetracycline is the most widely used antibiotic in livestock production in terms of animal feed additives. So that might be one reason why this particular domain of the overuse and misuse of antibiotics is associated with MRSA. The other reason, and this is right at the state of the art because I just had a conference call with people in the US, Denmark, and Belgium on this topic today, resides in this slide. It might be the result of 2 types of pressures, both antibiotic pressure and metals. And metals, of course, can be in the environment, and some metals are actually also added to animal feeds as micronutrients, such as zinc and copper. And that may influence resistance through co-resistance by shared mechanism or through co-selection. And this is very, very scientifically exciting, but perhaps from the public health point of view, a little alarming. And what this shows, here's a schematic of a bacteria here, and you see its cell wall. And within the cell wall, you see 4 embedded objects. And what they reflect are essentially proteins that can handle antibiotics or metals. So this is really what we mean when we're saying something is antibiotic resistant. It now contains, for example, on the upper left, a gene has been now expressed which permits the bacteria to pump out tetracycline. And that's a way of protecting itself against tetracycline, which will poison intracellular machinery. That same gene will also pump out cobalt. So that's shared mechanism. Similarly, down on the bottom, you see the CZC, and that is pumping out cadmium and zinc, and perhaps a little bit of cobalt. And what we now find is that that particular gene, that cadmium zinc resistance gene, seems to be found in the cassettes containing methicillin resistance from all the livestock-associated isolates in the Netherlands and Belgium. So maybe it's co-exposure to a metal which drives this system in a synergistic fashion. Or it could be what's represented here. And this is what we talk about co-selection for horizontal genes transfer. This is a transposon, which is one of those elements on a gene cassette, which can actually transfer genes, which then get taken up within chromosomal DNA and can be expressed. So this is the full machinery, if you will, by which horizontal gene transfer occurs. All I want to point out on this one is this is from Staph aureus, and this is within a cassette which encodes for TET resistance and also for methicillin resistance. And what you see, what I've circled there, are 2 genes that encode for mercury resistance. So what does it mean to have all of this on one cassette? It means that any one of these pressures will drive the movement of this cassette from one bacteria to another. So it doesn't have to be exposed to tetracycline, doesn't have to be exposed to methicillin if it's exposed to mercury. We feed a lot of food animals ground up fish. It's part of the wonderful diet we give them. Nobody's looked at whether the mercury that's going into their diet might contribute to this through this type of molecular machinery. Well, once again, here's our setting, our community setting whereby all of this is taking place, whereby people who are directly involved in livestock production and people who are bystanders or driving behind chicken trucks can also be exposed. And to end with our case study, why was Jane Doe coming in with community-associated MRSA in Lancaster County? She lives in Pennsylvania, remember, and she lives right there in that pink circle. Surrounding her are a large number of swine operations that have been permitted in Pennsylvania. So these are the sorts of coincidences that we really need to start investigating. And we need to use all the tools we have, epidemiologic, ecologic, as well as genetic, to determine what these risk factors are, even for people like Jane, who aren't involved in raising farm animals. So let me conclude now with some general comments. That first off, I've emphasized this issue of the agricultural use of antibiotics really for a pretty simple reason. You know, there's a famous bank robber in American history, Willie Sutton, and somebody asked him once, why do you rob banks? And he said, well, that's where the money is. So why do I look at animal livestock production? That's where the antibiotics are. So this industrial model of food animal production that we've developed over the past 40 to 70 years produces cheap and affordable food, but it has major costs. And I mentioned most of these are not incorporated into the costs that we pay at the grocery store, and certainly not incorporated into the balance sheet of the corporations or the wages paid to the farmers or the contract growers. In some respects, this model may reduce the incidence of food safety issues by a kind of economy of scale, for example, in terms of investments in food safety behaviors and practices, particularly at slaughterhouses, although I have to tell you, I'm not sure what those investments are, having started to study those. But the problem is, because of the concentration and the massive scale, that when there is a food safety problem, it tends to be larger because of concentration. And I think some recent events that have been occurring in Europe and in this country indicate that. In addition, the new way that we raise animals for food has introduced a new public health risk, and this relates to the enormous pressure, driving pressure from the overwhelming use of antimicrobials for resistance and pathogens, which is one of the major public health risks today. And there are strong associations between the use of antibiotics for growth promotion, that's GPA, and incidence of prevalence of resistant isolates in humans, both consumers, farmers, and workers. And there are a variety of pathways, both through the environment, person to person, releases from these operations, which contribute to the movement of antibiotic resistance originating within animal populations into human population, as well as just generally driving an expansion of the resources that microbes have to draw upon for resistance. And again, to conclude, that antibiotic resistance really is a critical public health crisis of our time. There was an editorial in the New England Journal some 3 years ago, which was entitled "The End of the Antibiotic Era." And I was interviewed last week by a radio station from Germany asking me what I thought would happen when we come to the end of the antibiotic era. And I said, I think we have some newsreels from that time, when more people died of infections than gunshot wounds in World War I. I think we know what's going to happen. We don't have to speculate. We overcame that very recently in our history, and we've been profligate in discarding all the advantages we achieved. So we really share an issue, which is how can we prevent or reduce our contributions? Remember, we're always up against the wall because these natural systems have been in existence for billions of years longer than us and have a knowledge of and the resources to deal with antimicrobial molecules. So how do we reduce our contribution to the increasing incidence and prevalence of antimicrobial resistance in human disease? And we have to do this fairly urgently because, as remarked upon by my good friend Ramanan Laxmanarayan, antibiotics really are an endangered species. We're failing to come up with new molecules. It's not so easy to come up with new molecules. And this is just some data, again, from FDA, which show the number of new approved antibacterial agents from the early 1980s through the mid-2000s. And you can see it's down to now about 1 or 2. These are not easy to come up with. So we're not generating new tools in our struggle against pathogenic infections. So how can we reduce the problems of community-associated MRSA, which is really the problem of MRSA? Many people have called for increased surveillance and data collection. MRSA is not currently a reportable disease in the United States, so we really don't have very good information. You notice I cited a lot of data from the Netherlands and other countries. It's because we really don't have a good idea of what's going on. The state of Maryland does not have good idea. The state of Pennsylvania has mandated as a guideline the collection of data at hospitals, but there's no systematic, unified, and publicly available compendium of those data. We really need to understand what we're dealing with. I think the application of molecular biology, as in the case of HIV, as in the case of tuberculosis and other major pathogens, can be extremely important in helping us understand places where these emerging strains are coming from and how they're moving through our population. And certainly, we need to phase out the use of antimicrobials in animal feeds. This is the paradigm instance of abuse and misuse of these precious drugs. So these are just some of the issues. I just want to point out that the knowledge that this use of antimicrobials in animal feeds was observed within 2 years after the practice began. So in 1949, the first use of antimicrobials in poultry feeds was permitted by the FDA. And in 1951, 2 veterinarians at UC Davis, in studying the poultry houses at the College of Agriculture reported resistance in poultry isolates from flocks that were receiving these drugs in feed. And they concluded with one of those really heartrending statements in the history of our field of public health. They said, perhaps in the future, public health should pay attention to this. But that did not happen for over 50 years. So I'd like to end by recommending some reading. The CDC website on MRSA is always good and has very good data and information on the latest information about community-associated MRSA. I refer to one of our papers where we really talk about this growing importance of thinking about this as a zoonotic disease and the notion of a one reservoir of both disease and pathogens. The sort of history of the evolution of resistance within Staph aureus was brilliantly described by Chambers and DeLeo, and goes way back in the history of the deployment of penicillin and other early drugs. And then the final one is probably the most recent summary of the different strains that are now occurring in animal species, and is one of the reasons why I suggest we shouldn't just talk about this as livestock associated. In fact, one of our students is looking at companion animals as potential sources and reservoirs of MRSA within homes. So we need to, once again, make sure that we have the proper critical perspective on this problem and not define it in the way that we have defined it last year. I have a lot of people to thank who've helped on this project, particularly Carol Resnick and Rhonda Jackson, without whom none of this could have been done, and the support that we've received from a number of foundations and federal agencies. So thank you.
Thank you, Dr. Silbergeld. And thank you all for coming. I do have a quick note for those of you who are watching online. If you have questions for Ellen, please first refresh your page. Just refresh your web page. We had some technical issues, but if you refresh your page before you send the question, that should resolve it. If you have already sent a question, you might want to refresh and resend. So while we're waiting for our online questions, we can open it up to any of you who are here in our live audience. Yeah, right here.
Hi, so I very much enjoyed your talk, and I very much enjoyed your talk, and I was very interested in one of the points you raised about a possible relationship between metals exposure and the continuing spread of MRSA. So antibiotic resistance is costly to bacteria to maintain. There has to be something or they'll lose the selection for the plasmid. But we've got so much antibiotic use and so many other exposures like metals in the environment that could be kind of forming a glass that you're pouring more MRSA resistance into from the CAFOs and other sources. I'm wondering, do you think that in your public health recommendation slide, another bullet point that might be useful is reduce metals pollution so that there's less support for this MRSA to take hold once it gets in the community?
Well, first off, you're right to point out the concept that there was always a cost of resistance, because otherwise, why wouldn't you find 50% of bacteria being resistant and 50% not if there was no particular reason or biological cost involved? And I think you are correct that we're dealing with a very different planet. I guess I really have to put it in those terms, in terms of the widespread use of antimicrobial drugs, speaking only of those. And the concept that for a community, and bacteria really work as a community, that for a community it may make more evolutionary sense in terms of preserving the species and enhancing reproduction to maintain resistance, at least the genetic machinery for resistance, rather than to, shall we say, mutate back to the susceptible phenotype or genotype. And so I think we are dealing with a very different circumstance, and that was partly revealed in studies that we did and others in Norway and Sweden did, that even after the removal of antibiotics from animal feeds. We've removed one antibiotic from animal feed, that ciprofloxacin from poultry feed. We didn't snap back to a position where all the pathogens found in poultry, for example, in the U.S., were now no longer resistant to ciprofloxacin. They've gone way down and are almost at zero now, but that's 7 years after the removal. So it takes a long time, perhaps, for the community to give up that resource. With respect to the additional value of constraining metal contamination, we still need to know a little bit more about this linkage before we would make that recommendation. Of course, there could be other public health reasons to constrain those exposures as well.
Thanks for such a great talk, Dr. Silbergeld. So multi-drug resistance has increased in a lot of other pathogens besides just Staph aureus in the last several years or decade. And not in a lot of different environments, but particularly within the paradigm of food safety and the food system. Do you think that the resistome of the modern farm is contributing to the resistance that we're seeing in those particular organisms? And if so, do you think that it's evolving along separate lines in separate species, or is there some degree of horizontal gene transfer that's conferring resistance from one species to another? I'm thinking in particular about Germany, where the E. coli is from an organic farm that uses no antibiotics, but the manure they're spreading on their crops is chock full of chicken and pig manure, which came from a CAFO or from some farm which used antibiotics.
Right, just on that last point, keep in mind that the use of animal waste as a fertilizer is considered part of organic farming, regardless of where the manure came from. I think it really is at the level of the resistome. I think dipping into the microbiome, that is, of all the organisms that might be present on a chicken, in a person, or on a farm, and pulling out one organism is a very limited lens, and it does not reflect what is the dynamism among the organisms within that microbiome. So I think the real next stage is to characterize the microbiome within, say, a farm, even within humans, and then within that microbiome, look for plasmids. Now, that is the molecular machinery that carries these gene cassettes from one organism to another, for the most part. Bacteria can take up naked DNA, but most of it is plasmid-mediated. And in fact, there have been studies done by Salyers and others to show that within a chicken farm, there is a buildup of plasmids within the soils where the chicken litter or waste is disposed. So we are, I think, enriching that resource. And then that becomes more generally available as all of this material moves around.
Any other questions here? We do have a few from our online audience. One question is getting to practices on the farms that have the animals where a lot of this seems to be initiating, and wondering whether if some of the manure was watered down using sprinklers, et cetera, so that the organisms didn't get aerosolized, would that diminish transmission?
The question of managing the waste material from the farms is a very disturbing part of this story. There are actually no regulations in the United States related to the handling, the storage, or the management or disposal of animal waste. Now, if you go to a wastewater treatment plant in your city where they collect a lot of biosolids out of human waste, those biosolids have to be tested very carefully, including for pathogens, before they can be released for any use, for example, as a garden fertilizer. There are no such requirements for animal waste. There are no requirements whatsoever for animal waste. And in fact, whatever guidance is given seems to be based on something other than empirical science, in that the USDA recommends that the chicken waste should be held for 90 days and then disposed of on land. One of our students went to a chicken waste pile and sampled it every week for 90 days and found that over that period of time, actually the number of bacteria increased. It did not decrease. and the number of drug-resistant bacteria, including Staph aureus, increased. How to manage all this waste? The aerosolization may be one part, but not necessarily the major part of the exposure pathway for humans. I think that the episode in Germany, for example, points out, and there was a similar one in the United States out in California involving spring onions, it's actually the routine use of these wastes as fertilizers or land amendments, and then that material gets on the plant or the crop and through the processing is not completely neutralized or removed. And that may be from just being in the soils. It's really a problem that starts both at the way in which we're confining animals in very large numbers, which then generates huge amounts of waste, and then the way in which those wastes are held and actually imposing regulations that are at least as stringent as the regulations we impose on managing human biosolids. I mean, otherwise we're really it's uncontrollable. We did a study in which we followed chicken trucks over in the eastern shore of Maryland, and by using air samplers within a passenger car, we were able to recover drug-resistant pathogens just driving behind a chicken truck. So we're not going to contain this problem if we wait until it's out. And spraying to stop dust may do something, but it's not the major problem.
Is there any proposed legislation that you know of at this point aimed at addressing this problem?
Not that I know of. The only concerns that have been, or only regulatory attention that has been directed towards this issue, that is the management of the animal waste. And just to give you an idea, we raise about 400 to 600 million chickens per year on the eastern shore. That is Delaware, Maryland, and Virginia. Each chicken produces about 2 kilograms of waste. So you can do the math and realize this is an enormous amount of waste material that has to go somewhere. And not all of that region is available to spread chicken waste on. There are towns, there are cities, there are beach resorts, golf clubs, etc. So it's really going into a very small area and overloading those areas. The big concern, as expressed by EPA, has related to impacts on surface waters from over-nutrification, that is, eutrophication through nutrients such as phosphorus and nitrogen in the waste. There's absolutely nothing said about the presence of either antibiotic drugs themselves or drug-resistant bacteria, and then the issues related to odors, which are certainly pretty powerful if you've ever been near swine farms and poultry farms and waste impoundments, but again, no attention given to pathogens.
There's a question about whether certain types of antibiotics are more of a problem than others, such as whether inhibitory versus CIDAL or CIDAL antibiotics are a problem.
Well, in general, I would answer the question by asking you to keep in mind the notion that antibiotic resistance genes tend to travel in groups. And there has been legislation introduced in the U.S. Congress to remove what are called critically important antibiotics from animal feeds. But the problem is that there could be a drug, let's take tetracycline, for example, which has been so used and abused over decades that TET resistance is widespread. And tetracycline is no longer really considered a critically important drug. Yet some of the data indicate that it's a TET resistance gene that might in fact participate in the movement of methicillin resistance. So if we expand our thinking beyond one bug, one drug, we really see that we've got to come to grips with this problem in a holistic manner.
Okay. And we have a question from Miami-Dade County, wondering whether we can be confident that organic poultry or meat in general is free of antibiotics.
I believe number one, in most instances, the meat product that you get in the grocery store will have very low or no detectable levels of antibiotics. That is, that the only regulation that the FDA has put into place, frankly, has been to ensure that for certain longer-lived drugs, there has to be a period of time before slaughter where they don't get the drug. But as you know from clinical or veterinary medicine, you have to take your antibiotics every day. They don't stay in the body that long. They're washed out fairly rapidly. So exposures to antibiotics are probably fairly low within the food supply, with one exception, and that would be the use of arsenicals, which I understand the drug company has just announced they're no longer going to make the drug, so perhaps we won't see that anymore.
Okay. Do we have any other questions here from the live audience? Before I go on, we do have another question from online. Wondering whether this is a problem in both organisms with a cell wall and without a cell wall.
Well, I'm referring to bacteria here.
Okay. And I think that is all that we have in terms of questions, unless anyone here has any more. I think that wraps it up for today's presentation. I really want to thank Dr. Silbergeld so much for her time and wonderful presentation, and hope that you'll all join us again next month for our grand rounds in July. Okay, thank you very much.
Thank you.